So let's get into the principles of tree building and phylogeny in general, because these are interesting. So the main principle is going to be our principle of parsimony. Parsimony, for those that don't know, means basically cheapness. Yeah, the least number of evolutionary steps. It's like an Occam's razor kind of thing. If we went out to dinner and at the end of dinner I pretended to reach for my wallet but didn't actually, and said, you know what, yeah, I think you can get this dinner, I wouldn't be being cheap. I would be being parsimonious. Yeah. In spending as little as I can to get the result. So it's just good science, I'm afraid. Yeah. Right. So we want to. So we can't ever find what was the true path of evolution, because you could have an A nucleotide that turned to a C, that turned back to an A, that turned to a C, that turned back to an A, and then eventually turned into a G, and then stayed as a G for the rest of time. But we can't reconstruct that path using parsimony-based methods. What we would say is the cheapest way to get from A to G is to make one change, A into a G. And so we know we're probably going to be underestimating the amount of evolution that has happened there, because we can't really overestimate it. So yeah, we are almost certainly underestimating it. And so that's what most simple phylogenetic trees are. They're based on parsimony. And the benefits of this are that you can whip out these trees really quickly and with zero effort. The bad part is that they sometimes tell you things that you can say, boy, I know that's not right, and show you relationships that just really aren't there and that more sophisticated methods would exclude, or at least they are less likely. All right. When constructing trees with parsimony, it's very important to root the trees. So you can see up here on our little tree of life, there we have a root. And that's great. So the root is the original ancestral condition. So this is where the last common ancestor of all these things. And remember, it doesn't have to be a single ancestor, and for species like us where it takes more than one to reproduce, it pretty much has to be a pool of ancestors. But yeah, the one or ones that contributed to all of the genes that are there. So what you need is a thing called an outgroup. So an outgroup has got to be homologous. It's got to have the same features, but you want the outgroup to be related to itself, the other members of the outgroup, and you want your in-group, like the thing you're studying, to all be related, and you want them to be close but not identical, so that you will get a tree that has, let's say they used bacteria as an outgroup here. So they reconstructed a bacterial tree, there it is, and then they reconstructed an archaeal tree and a eukaryotic tree, and then they reconstructed all these together. So they knew that eukaryotes and archaea are related, and so the point where that branch meets the bacteria branch, right there, bam, that's where they put the root. When you're looking at a tree, what you have to remember is that if it is an unrooted tree, so if they haven't done careful work to establish where the root should be and shown that with math and numbers and science, then really any topology is possible. So it's equally likely that the root is right here between spirochetes and proteobacteria, and that everything else is a descendant from that. So you can basically mentally rearrange these trees with any one of these nodes as the potential root, unless you have good statistical basis for assigning a root to a tree. This is a mistake that a lot of people, even scientists working in fields where they should know better, will make. Yeah, so it's worth paying attention. So yeah, outgroup is homologous but different and related to itself. You want multiple members in the outgroup because you want them to group together, and you want to see where they attach to the rest of your tree, because that point marks the root of your tree, wherever it may be. There you go. You have been enlightened. Yeah. Okay, Yeah, there's a lot of ways that genetic change can contribute to evolution. It's not just gene drift, like mutations happening. That's one of the less likely things to contribute to species level evolution. You will often get gene duplications within a genome, very common. You will get gene deletions. Oh my gosh, the human genome has so many duplications and deletions in it. It's ridiculous. And you'll get shuffling, where you get genes sort of swapping parts. In this case, it's exon shuffling, but it can happen. This is like when your mom and dad loved each other very much, et cetera, and the chromosomes were coming together in order to make those little gametes. They would do that little switching, and so you get a lot of exon shuffling from meiotic processes. All right, so horizontal gene transfer, we already mentioned, it is movement of a gene from one organism to another organism. Both have to be on the Earth at the same time for this to work. Yeah. And so like when a virus comes in and infects a cell, the virus will stick sometimes its DNA into the cell. So bacteriophage, like this little guy, looks like the T4 phage, will do that. But also viruses like HIV do this. Also, bacteria pick up resistance genes all by themselves with nobody injecting anything. So here's our little plasmid, through conjugation, for example, or just sort of slurping up DNA from some other dead cell. Lots of different ways for horizontal gene transfer to happen. So that's how you can end up with a gene that is homologous but is not necessarily following the same evolutionary pattern as the rest of the genes that make up the genome it's in, if that makes sense. Okay, fine. So mutations, if they don't change if it's a DNA mutation that does not change the coding, quite often we'll say this is selectively neutral because the gene only exists to produce the protein, and the protein exists to do a job. And if you're making exactly the same protein with slightly different instructions, it's still exactly the same protein. And chances are you will not have any sort of phenotype. And so we call these silent mutations, or some people call them neutral mutations. They're not all neutral. There are silent mutations that affect splicing, that affect regulatory sequences, that affect promoters and terminators and enhancers. And all of these will have gigantic effects on expression of a gene, maybe more so than just mutations in the gene itself. So it's worth noting. Okay. Misalignment, you can get duplication. That's a thing that can happen. Duplicated copies. Yeah, the genes that make up hemoglobin are copies of an alpha and beta chain. And the alpha chain and the beta chain are real similar to each other because they are from a gene duplication. So somewhere out there is an ancestor of a single globin chain that can bind a heme group. And these have been identified. They're found in other organisms, for example. What we have is 2 copies of those that will then assemble into a little bundle of 4 with 2 copies of each version. So there you go. All right. Sometimes you get duplication of the entire genome. Very common in, for example, commercial wheat, corn, crops like that. Plants will often have, they'll have extra sets, full sets of chromosomes. Generally, in people anyway, you do not see this, but there are certain kinds of mammals. For example, platypus and echidna have many, many sex chromosomes. I forget if it's like 5 or 7 or 9 X's and Y's. And they're basically copies of one ancestral version, but they've just kept doubling them up and carrying them around. And now everything has evolved to the point where they probably have to have all of those things. There's probably no way to untangle that particular knot. So it can happen to some extent. And it does happen in evolutionary time. For example, people have most of the same genes as mice, but as you'll see in a couple slides, the genes are rearranged on different chromosomes. You'll have a whole chunk of genes that will move from one chromosome to another through evolution. And sometimes that'll kill you, but sometimes, you know what? It's okay, and you get through it, and everything's all right on the other side. Yeah. Yeah, a lot of eukaryotic genes come from exon shuffling. Prokaryotes don't have exons, they just have genes that don't have any junk in the middle of them. But the exons, of course, give us a little more flexibility, is what we like to say. I feel like that's trying to make ourselves feel better for having all this junky DNA that isn't very easy to analyze. But that's my personal bias, coming from a splice-free world of viruses for the most part. There are also transposons, and these are little sequences that are able to insert themselves into DNA, cut themselves out of DNA, and jump from one place to another within DNA. I forget the statistic. It's something like 1 in 300 births, something like that, or maybe it was 1,000 human live births. You would have one of these transposon jumping events. Our genes are crawling with lines and signs and retrotransposons and full retrovirus genomes. We have far more of that sort of thing than we have of our own genes. So yeah, this stuff is going on all the time, and thankfully, it usually doesn't kill us. Yeah. One of these transposable elements in humans is called the ALU sequence. Yeah, I'm not going to test over that. Just if you see it come up somewhere, you'll say, "Oh, I remember that. I remember that from bioinformatics class," and you'll be right. Okay, I think this is a good point to pause, and then we'll come back and get into this a little more.